we'll move to the next talk. And this is going to be Dr. Husseini from Iran, and he's going to talk about the anti-proliferative effects, cell cycle alterations, and apoptosis induction of DDW on Z470 and HD29 human carcinoma cell lines. Dr. Husseini?
Yes, thank you. All right. Actually, I must confess that I have learned a lot today because I have been exposed to too much diverse information on DDW. As I have mentioned last night, we have been in this business for the last couple of years, and from when we have had access to this DDW. And we have invited medical universities and to work on this DDW. And we have ordered some sort of research activity to the Tehran Medical University, to Dr. Ibrahim Azizi's lab. Actually, I'm presenting his slides. He couldn't be with us today. The work that we present here is the anti-proliferative effects, cell cycle alterations, and apoptosis induction of DDW on T47D breast cancer and HT29 colorectal cancer cell lines. I don't need to mention really that how treatment of cancer is important, and that the cell cycle alteration and or decreased apoptosis induction following chemotherapy are primarily due to molecular changes of cancer cells. Studying new compound alone or in combination with potent chemotherapeutic drugs such as doxorubicin can open new strategic approach in cancer therapy. Previous reports indicated that the potential anti-cancer effects of deuterium depleted water, mainly from Dr. Sorry, I forgot. I forgot your name. Shumlai. Is that right? Yeah, okay. Okay, so we have been motivated to study the effectiveness of this DDW water in cancer therapy. And we have done this because we have, as I said, we have had access to two types of DDW. We called them A and B. And those days we didn't know which one is what in terms of the content of deuterium. So we have done these cytotoxicity effects, cell cycle alteration, and apoptosis induction studies on those cell lines that I have mentioned in the presence or absence of doxorubicin. In terms of methods, all of those solutions, buffers are just prepared based on what have been explained in literature. For the cytotoxicity experiments, we have used 10,000 cells or for the case of the colorectal carcinoma cells, 5,000, cultivated in 96 well plates. The culture media replaced after 48 hours with DDW A or B in the presence or absence of 250 nanomolar doxorubicin. Cells were then counted at 540 versus 690 nanometers using a sunrise plate reader after treatment with MTT in days 2, 4, and 6 for T47 or at 10 days for the HT29 cells. And all of the experiments were conducted at least 3 times, and the results are demonstrated as means plus and minus and standard deviation. For the cell cycle alteration, we have used 40,000 cells from each, cultivated in 12 well plates, and the culture media replaced after 48 hours with DDW A or B in the presence or absence of 40 nanomolar doxorubicin. Cells then treated with trypsin EDTA washed with PBS buffer, centrifuged and homogenized a DAPI cocktail and incubated for 30 minutes in dark. Cells then transferred to flow cytometry tubes and the fluorescent intensity read and recorded at FL4. Data analyzed and the percentage of cells at G0, G1, S or G2M phases defined using FluMax program. And for the apoptosis experiments, we did exactly the same, but we treated the cells a little bit different. And then when we transferred the cell for flow cytometry, and the fluorescent intensity read at FL1 versus FL2 and recorded for each quadrant.